And then I watched the movie by Meryl Streep called First Do No Harm. I never thought Meryl Streep did a movie about the ketogenic diet. And I watched this and I realized that this is for real. And I immediately went to PubMed and I just went down the rabbit hole and I read literally paper after paper for a couple weeks. And I put my hands up and I said, this is what I'm going to study.
Charge up your axons, ready your receptors, and shift your lobes into upper beta phase. You are listening to Smart Drug Smarts, the podcast dedicated to helping you optimize your brain with the latest breakthroughs in neuroscience, nootropics, and psychopharmacology.
Hello and welcome to Smart Drug Smarts. My name is Jesse Lawler. I am your host, and this is the 147th episode of this podcast dedicated to the ongoing betterment of your brain, including by radically changing its fuel sources from its traditional glucose to good old-fashioned fats, or the breakdown products of fats, something called ketone bodies. This is an episode that's been a long time coming, probably long overdue, but in many ways, the whole reintegration of fats as a healthy part of the human diet is probably long overdue. We've been, as a culture, probably unfairly demonizing fat for the better part of the last 30, 40 years. And luckily in the past 5 years, that's really started changing. And on the forefront of some of the realizations that are coming around as to the usefulness of fat within the human diet, both for therapeutic and performance purposes, is our guest today, Dr. Dominic D'Agostino from the University of South Florida and also part of the Florida Institute for Human and Machine Cognition, which is an institution that's kind of a friend of the Smart Drug Smarts family. We've talked with a couple of Dr. D'Agostino's colleagues over there, Dr. Ken Ford and Dr. Don Kernagis in some previous episodes. But anyway, this one is going to be all about ketosis and the brain, all about what happens when you give your brain fat as a fuel source. It's a deep topic. There is lots to be said. This is a longer than usual episode with a longer than usual interview. If you hang around until the very end of the episode, I will tell you about some recent scientific findings as to when it is best to exercise insofar as helping your memory. Exercise, famously good for all kinds of things. Memory is not necessarily what we think of first, but when you time your physical exercise versus the time of the learning of the things that you want to remember apparently has some results. And we will find out what those are in the ruthless listener retention gimmick. But first, as usual, let's kick things off with This Week in Neuroscience.
Smart Drug Smarts, This Week in Neuroscience.
So humans have big brains. We all know that. It's sort of our species birthright, our bragging right for being humans. There's not a lot of other things that we're completely exceptional at. We build tools, but some other animals do that. We sweat particularly well. We sweat in all parts of our bodies almost. Not a lot of other animals do that, but our brains are really our claim to fame. I remember when I was a little kid and I was really into dinosaurs like most little kids, and there was this statistic that was always trotted out in my kids' dinosaur books, how a stegosaurus, this massive dinosaur that, is like the size of a couple of water buffaloes put together, had a brain the size of a walnut. How could that be? How stupid was this little thing? And of course, it makes sense intuitively that intelligence has something to do with brain size. And yes, humans do have conspicuously large brains, even compared to other mammals and even compared to other primates. So the intuitive suspicion, and there's certainly something to this, is that brain size probably has a lot to do with intelligence. But that can't be the end of the story because there are animals with bigger brains than humans. Elephants have much larger brains, and elephants are alleged to be pretty smart, but nobody seems to be saying that elephants are as smart as humans. And there are many species of whale that have much bigger brains than humans. If you were to pull them out, put them on a scale next to us, measure them by volume, any of those things. And yet here we are, demonstrably smarter than anything else. So what's going on? A new paper put out by researchers at the University of Adelaide in Australia and the University of Witwatersrand in South Africa have come up with a really interesting new wrinkle in this theory, and it has to do with our evolutionary ancestors being particularly bloodthirsty. And not bloodthirsty in the way you think, but that our brains are actually taking in blood at a faster rate, metabolically speaking, than would be expected by an animal with our brain size. In other words, according to this theory, a given cubic centimeter of human brain is using up blood, turning oxygenated blood into deoxygenated blood and kicking it back for more at a higher rate than another cubic centimeter of brain would be at any other species you could find. Now, how would they know this? And how would we in fact know what the comparable amount of blood usage in a brain is in some of these human ancestors, the Australopithecines and species like that haven't been alive for the last couple of million years? Well, it turns out that that's where skulls come in. So if you've ever looked at an animal skull, there's that one really obvious hole at the bottom or at the back, depending on the type of animal, where the spine connects. It's kind of the major structural connective point, and you see a big central hole there in a skull. But if you look closely, you also see a couple of little smaller holes off to either side of that primary spinal hole, and those holes exist to allow the passage of what are called the internal carotid arteries, ICAs, which deliver the lion's share, like the massive supermajority of blood that makes its way to the brain, travels through those arteries. And so the scientists thought, hey, if we look at what's called the lumen radius, which is essentially the area, the size of the hole, of these skull holes to accommodate the internal carotid arteries, we can see how the size of these holes has changed relative to the brain size and other things that we know about the animals, both human ancestors that are extinct, primates that are alive now, and of course modern humans. Now you might say that that doesn't necessarily hold, because what if the blood's just flowing faster? You could think about a garden hose and think, well, you could have either water flowing really quickly through that garden hose or water kind of trickling out. Either way, the hose is the same size, so how do you know how much water is actually flowing? But it turns out that it does not work that way when it comes to arteries. This measure of width, called the lumen size, and the wall thickness of large arteries are dynamically controlled throughout life by blood flow requirements and blood pressure. And these relationships are so well understood by science that they're considered to be laws. They've got formulas where you plug in things like blood pressure and wall thickness, and it will basically tell you how wide the artery is going to be. And they've tested this experimentally in animals. They actually did a study where they reduced the blood flow of a common carotid artery by 35% in a rat, and over the course of several weeks, the inner radius of the artery decreased according to what was expected by these formulas to within 11% of the theoretical value. So there's pretty good reason to suspect that the size of these holes in the skull really is proportional to the amount of blood that's flowing to the brain. So whereas with most animals, looking at the ratio between brain size and the size of these holes for the internal carotid arteries, as the animals get bigger, as the animals' brains get bigger, of course the holes typically get bigger, but they get bigger at a ratio less than 1. So if you double the size of the animal, you don't necessarily double the size of these arterial holes. What you typically see in most animal species is a ratio a little less than 1, somewhere between 0.8 and 0.9. But looking at human ancestors, things like the Australopithecines and Homo erectus, Homo habilis, and that fan favorite, the Neanderthal, what they found was really different. They see a ratio of over 1. So as modern Homo sapiens were developing, as our brains were growing in size, we had that going for us as far as our ability to think and process information, but our ever larger brains were also consuming oxygen from our bloodstream at a higher and higher rate, and putting that oxygen to use for the brain's metabolism. Synaptic transmission, basically one neuron talking to its neighbors, accounts for more than 1/2 of the brain's total metabolic energy requirements. So turning that on its head, we can infer that if there are higher metabolic energy requirements, there's more synaptic transmission going on. Summing up their findings, the project leader, Professor Emeritus Roger Seymour from the University of Adelaide, said, "Brain size has increased about 350% over human evolution, but what we found is that blood flow to the brain has increased by an amazing 600%. We believe this is possibly related to the brain's need to satisfy increasingly energetic connections between nerve cells, which allowed for the evolution of complex thinking and learning."
Smart Drug Smarts, the podcast so smart, we have smart in our title twice.
Picked up a 5-star review on iTunes from listener Melanator, who said, "Excellent work. This podcast reliably provides a comprehensive yet colloquial supply of information for more than the average consumer. Thank you, Melanator. Much appreciated. And thanks to everybody that helps spread the word on Smart Drug Smarts. I know that a lot of you out there are being good little Johnny Appleseeds and flinging around links and mentions and things like that, all of which are very much appreciated. I got one email from somebody this week just back from Burning Man. I assume everybody knows what Burning Man is, so I'm not going to go into an explanation of that here, but somebody that had brought Nexus, our cognitive stack over at Axon Labs, he said that 6 nights out of 7 he was clean and sober at Burning Man, but in order to stay out late with his friends and have lots of mental energy, not be relying on things like 2 a.m. espressos and cans of Red Bull, Nexus was his go-to. That's our cognitive stacks based around aniracetam, has CDP, choline, and a couple other things. I was super happy to hear that. I've been to Burning Man. I didn't go this past year, but very happy that our Axon Labs products made it out onto the playa. Nexus and its sister stack, Mitogen, are available online over at axonlabs.io. And if you wind up trying either of those stacks in any interesting environments, or just using them as you're participating in interesting, cognitively demanding tasks, I would be super curious to hear what you're up to. If it's photogenic, drop a photo. If it's not, just shoot me an email. My email is jesse@smartdrugsmarts.com. Another way to reach out to us is on Twitter. We've got all the social media stuff that you would expect, but I'm mentioning Twitter particularly here because the guest in today's show, Dr. Dominic D'Agostino, he's actually going to be doing a live Twitter event with us this coming week, Wednesday the 28th of September. We're calling this an Axon Chat, where anything you want to ask him about ketosis, ketogenic diets, fasting, he will be around for a live Q&A. You can check that out online at smartdrugsmarts.com/AxonChat. That's AXONCHAT for details. But if you're on Twitter already, it's going to be super easy. You just show up at the right time. Check out that link for all the details. And while you're on the Smart Drug Smarts website, if you have not yet done so, we do have a newsletter. It's called The Brain Breakfast. It comes out about weekly, and you can sign up for that at smartdrugsmarts.com/newsletter.
Smart Drug Smarts.
So I'm about to be speaking with Dr. Dominic D'Agostino, as mentioned, from the University of South Florida, who is something of a ketosis celebrity, if there can be said to be such a thing. And I think there probably can. Ketosis is one of these things in ketogenic diets that is exploding in public awareness and popularity right now. It's resurgence sort of coming hand in hand with the increasing idea that we've sort of had it all wrong culturally, thinking that grains and carbohydrates at the bottom of the food pyramid is the be-all, end-all to the way that human beings should eat, and that fats are inherently bad and should be demonized and minimized within our diets. Those ideas, which have been sort of cornerstones of nutritional dogma for the past 30, 40 years, have come increasingly under attack, in large part by the work of people like Dr. D'Agostino, looking at some of the pieces that didn't quite fit in that story, things like high-fat diets being very, very helpful for people that have severe epilepsy and treating some of those conditions. Why is that? Is this some sort of weird physiological fluke? Why would what a doctor or nutritionist in 1990 would have told you is like the worst diet of all possible diets, why would that be the most effective way of treating juvenile epilepsy? So we're seeing sort of a massive phase shift in the way that we think about fats within our diet. Are all fats the same? Are all carbs the same? How much do macronutrient ratios matter? Is that a red herring? How can some of these previously unpopular foods be used therapeutically? How can they be used for performance enhancement purposes? And how might they be used by just a middle-of-the-road person looking for a regular healthy diet? These are questions that are far from settled, but they're questions that are getting publicly asked now in a way that seems more open to a spirit of inquiry than would have been the case 10, 15 years ago. So we're going to be talking a lot about ketosis, and I feel like we're obligated to kind of give a quick rundown of what that is before we dive too far into it. I would really recommend, if you're completely unfamiliar with the concept of ketosis, maybe jumping back to Smart Drug Smarts episode number 56, when I talked with Dr. Thomas Seyfried about ketosis versus cancer. But the highlight reel version is ketosis is the state that your body goes into when it does not have enough blood sugar to run on, doesn't have enough glucose circulating to take care of its physiological needs. It's scraping the bottom of the barrel on glucose, and it needs to find an alternate power source. And as you know, our alternate power source within our body is fats. So in a starvation situation, this would mean going into our adipose tissue, going into the fat stores that we have within our body, cracking open the canisters and releasing some of these fats or the breakdown products of fats into the bloodstream and starting to use those as a secondary fuel source. Some of the breakdown products of these fats are called ketone bodies, and thus the term ketosis. Now, a ketogenic diet is when you're not necessarily relying on your body's fat stores to give you fat, but you're actually eating a high-fat diet, so you're getting nutritional ketosis. Basically, you've got fat coming in nutritionally that's matching the fat that's going out energetically, so you're maintaining a physiological equilibrium. You're not dipping into your body's fat stores. You're not necessarily building them up either. But in the case of a ketogenic diet, ketosis is a long-term lifestyle rather than something that happens on an emergency basis because you're starving. Now, it's kind of amazing to me that we have this entire backup power system that works as well as it does and is just part of our normal physiological evolutionary inheritance. That's amazing. But maybe what's equally amazing is that in some ways, our bodies and our brains in particular seem to actually function better. That's not a blanket statement. There's obviously a lot of fine-grained details here, and that's what we're going to be going into in this interview. But ketosis is a really, really deep topic. Books can have and certainly will continue to be written about it, so there's more to be said than we can fit in a one-hour interview. But in this episode, we're going to be focusing on ketosis in the brain, particularly what ketosis does to our brain states, perceptually as far as long-term brain health. Are there optimal ways to do ketosis, and basically how can we take advantage of this? So as warned, as promised, this is a longer interview than usual, but it is chock full of good information, and it does get pretty scientifically jargon heavy at some points. Oftentimes when doing these interviews, I'll try to interrupt the guest and like, hey, can you define that term? And a few points in here I did this, but after a while I kind of realized Dr. D'Agostino, he talks science, and he's throwing a lot of vocabulary all the time, and I just didn't want to constantly be interrupting the flow of what he had to say to define terms. Luckily, he's very clearly spoken, so if you need to look up a word or two every now and then, at least you can probably sound out what he was saying. But you're going to hear terms like hypovolemic, which, by the way, means of or pertaining to a low blood volume in the circulatory system, or bolus, which is a single relatively large quantity of a substance, such as a drug, intended for therapeutic use and taken orally. And that's the type of terminology you're going to get a lot of in this conversation, so just be aware, brace yourself as necessary, but I think even amidst a big stew of vocabulary, some really important ideas still come across loud and clear. So with no further ado, let's chew the fat with Dr. Dominic D'Agostino.
I went through a nutrition science undergraduate program at Rutgers University, just fresh out of high school. And the consensus there was that the ketogenic diet was sort of this fad diet, something of self-help books, I guess you could say, and really put a negative stigma to it. But they didn't even cover the fact that the ketogenic diet, the actual origin had to do with, it was a powerful metabolic therapy for drug-resistant epilepsy. I mean, that's really how it evolved. And then Atkins sort of popularized it for weight loss. So my early views were sort of negative, and I went through a nutrition science program. I was taught carbs are good, fat's bad, all that stuff. And I didn't pursue a PhD in nutrition just because the options were very slim. It's like, go work for Kraft or go work for General Mills or something like that. I wasn't really interested in that. And I was more interested in physiology and neuroscience. So that's what I went and did my PhD in neuroscience and physiology. And then I did patch clamp electrophysiology, which is measuring action potentials and neuronal activity in different cellular preparations, whether it be a brain slice or cultured neurons, some EEG work. And I was very tuned into the brain as being a very metabolically demanding organ, very high metabolic demands for its size. And I was actually looking at glucose and lactate, and alpha-L polylactate was something that I used. I was into mountain biking at the time, so I was interested in using that as an alternative energy source. In particular, in the context of hypoxia and ischemia.
Just to give the terms there, hypoxia is lack of oxygen and ischemia is what?
Ischemia is sort of both, like a lack of substrate, really, and a lack of oxygen and glucose to the brain. So yeah, my PhD was on the neural control of autonomic regulation, so sort of how the brain controls our respiration and physiology and autonomic functions in the context of hypoxia. So in the context of a lack of brain blood flow or a lack of oxygen, stroke, brain injury, things like that. So I kind of had an understanding and an appreciation for the substrates that nourish and energize the brain. And it wasn't until I finished my PhD and did my postdoctoral fellowship with Dr. Jay Dean at Wright State University, who was funded by the military, I hit on a grant from the Office of Navy Research to study oxygen toxicity seizures and to look at the cellular and molecular mechanism of oxygen toxicity seizures. And we developed a lot of unique technologies, including hyperbaric atomic force microscopy and confocal microscopy. And these instruments allowed us to actually look at the mitochondria under situations that would mimic, for example, a Navy SEAL diving with a closed circuit rebreather. So if you can envision cells exposed to high levels of oxygen and using various tools that allow us to look at reactive oxygen species production, membrane potential, mitochondrial function, things like that. And I became much more appreciative of metabolism in that context. I'm in a pharmacology department and physiology department, and I stumbled across, some people that had an influence on me, including Mike Dancer. He's an epilepsy patient, actually in the UK, and he had communicated with me on a nutrition board, and he had tried many different drugs to control his epilepsy and stumbled upon the ketogenic diet. And Mary Newport, she's a pediatrician here and made popular the use of coconut oil for various things, including Alzheimer's disease.
Yeah.
And her husband had advanced Alzheimer's disease, and he responded remarkably well to coconut oil and medium chain triglyceride oil. That caught my attention. And then I became more interested in, the ketogenic diet and ketones. And then I discovered the Charlie Foundation. It's run by Jim Abrams. His son was Charlie. And he discovered the ketogenic diet and worked with Johns Hopkins Clinic and was able to get his son's seizures under control when no drug was able to do it. And he became a very vocal advocate of the ketogenic diet, arguing that it's grossly underutilized. And then I watched the movie by Meryl Streep called First Do No Harm. I never thought Meryl Streep did a movie about the ketogenic diet. And I watched this and I realized that this is for real. And I immediately went to PubMed and I just went down the rabbit hole and I read literally paper after paper for a couple weeks. And I put my hands up and I said, this is what I'm going to study.
All right.
I'm studying CNS oxygen toxicity, which is drug-resistant seizures. And it's a limitation of hyperbaric oxygen therapy. But more importantly, from my funding agency's point of view, it's a limitation for Navy SEAL diving safety and performance. So I was like, okay, let's figure out what this diet is, optimize it, and maybe figure out some tricks where we can develop exogenous synthetic ketone molecules to further augment the therapeutic efficacy of the ketogenic diet.
And increasingly, the last couple of years in the blogosphere, you start seeing people talking about it for athletic performance, particularly things like ultra marathoning and some of these long distance type sports. What I'd kind of like to talk about with you, because I haven't heard as much about it, is is what ketosis can do for a person's cognition. I very much suspect that the answer is going to be a lot. The types of changes that we're seeing in the brain as far as stopping the electrical positive feedback loops that cause seizures and epilepsy, there's obviously some pretty profound effects going on. What else do we know about how it's affecting our brains?
Yeah, that's a topic that I'd love to discuss. I have kind of a personal opinions and anecdotal data, and then from the colleagues that I'm working with, they have some interesting data. And we also have an Alzheimer's study going on with a ketogenic agent. It's double-blind, placebo-controlled study, so I don't know the results yet, but could definitely speak as to the benefits that I think people could get, and not just from the ketogenic diet, perhaps exogenous ketones too.
Maybe to start off with just a bit of terminology, and I do not know the answer to this, but ketone bodies are basically 3 different water-soluble molecules. It's, correct me if I'm pronouncing any of these wrong, but it's acetoacetate, beta-hydroxybutyrate, and then acetone. Is there any practical reason for somebody that is interested in getting into ketosis thinking of those as 3 separate things, or can we kind of think of them collectively as ketone bodies and leave it at that?
I think it's good to just call them ketone bodies, and of course, ketones are kind of ubiquitous in nature, right? We have raspberry ketones. The ketone bond and molecule is kind of found throughout nature and our bodies, but ketone bodies were named by a physiologist, and they are very specific to the molecules that you just mentioned: acetoacetate, beta-hydroxybutyrate, and acetone. And acetone is a spontaneous breakdown product of acetoacetate. So our body makes beta-hydroxybutyrate, I would say, is the primary ketone molecule. The hydroxyl group makes it a little more stable and is elevated like in about a 4-to-1 ratio of beta-hydroxybutyrate to acetoacetate in the blood. And sometimes you see people with like a 2-to-1. So we take this into account, that there is fluctuations in the ratios of these two depending on the enzymatic and tissue sort of distribution and utilization. For your mitochondria to use beta-hydroxybutyrate, it must convert it first to acetoacetate, and then it kind of splits and goes to acetyl-CoA ultimately, and part of the Krebs cycle. And acetone's kind of interesting too. It's not really used as a metabolic fuel, but it has a lot of subnarcotic concentrations of acetone. Of course, acetone it's a nail polish remover, so it could be dangerous at high doses, but low levels can impact brain homeostasis in many ways. And I think Gary Yellen from Harvard actually did some nice work showing that it opens potassium channels, helps hyperpolarize the membrane potential to offset glutamate hyperexcitability or glutamate excitotoxicity. So it can work in that way. And so there's a lot of different ways that these ketone bodies can function. And I think when it relates to seizures, it's important, we found in our experiments, that if you just spike beta-hydroxybutyrate really high, you don't get the neuroprotective anti-seizure effects, at least against oxygen toxicity seizures.
Yeah.
But I realized I had to go back to the drawing board when we first tried a monoester of beta-hydroxybutyrate and other things that elevate beta-hydroxybutyrate. And then I stumbled upon the work of Henry Bruninggraber from Case Western, and he had developed various monoesters and diesters of acetoacetate. And what was interesting is that it elevated both beta-hydroxybutyrate and acetoacetate in more or less a 1-to-1 ratio. So you had both of them elevated. And, we screened and tested a lot of things, and it was the ketone esters that elevated both of the ketone bodies, acetoacetate and beta-hydroxybutyrate, in a specific ratio that conferred profound anti-seizure effects in our model. And then I went to the literature, and it was actually very clear in the literature, if I would have studied a little bit more and talked to different experts in the field. Jung Rowe, who is really a leader in understanding mechanistically how the ketogenic diet works. He's a pediatric epileptologist, and he was like, yeah, the balance of the data indicates that you need to elevate acetoacetate. That's really important for seizure control. And when we did that, it was remarkably effective. So we have been developing various ketogenic agents that elevate the 3 ketone bodies in different ratios and applying them to different disease pathologies. Now we're looking at things like anxiety and cognitive function and also performance. So beta-hydroxybutyrate is probably the ketone body for brain energy metabolism, but it's clear that acetoacetate is also a major contributor and has some unique anti-seizure neuroprotective effects. So I think elevating just beta-hydroxybutyrate is not a good idea. I think you need to kind of pay attention to nature, because nature balances these things out with various interconverting enzymes, and then look at those ratios and try to mimic that, and even maybe try to elevate one a little higher over the other depending on what kind of physiological or therapeutic effect you're looking to produce.
With a lot of stuff that's sort of hot right now in the health community, things like the paleo movement and ancestral living, there's a lot of people out there putting forward the idea that living in ketosis, eating a ketogenic diet, is a lot more normal as far as what we were evolved for than the type of higher glycemic index diet that we're typically eating nowadays. Where do you stand on this? I mean, how much of a person's life or, percentage of a person's year do you think would have been spent in ketosis back in a pre-agricultural era, 10,000, 50,000 years ago, whatever it is? Is this a real ancestral way of eating?
Yeah, I think so. Before, when I got into studying the ketogenic diet, I pulled a lot of books on the origin and the evolution of human diets and, from mostly like very academic circles. And a lot of it wasn't really promoting a low carb or a ketogenic diet. But what I garnered from reading the research was that the human diet had a lot of fiber in it and very little processed carbohydrates that would produce the type of glycemic response we see now with the foods that are on the market. And it was very rare to stumble upon fruit that was ripened or to be courageous enough to knock down a beehive and then eat all the honey and things like that. So it's pretty rare. And it's also relates to our geographical origins too. So depending on what part of the climate, and people didn't move around like they do now. So I think we are more or less in some ways hardwired for certain types of diets to some degree. I think you could easily make the argument that the Native Americans have well over 2 times the risk of type 2 diabetes, are not well adapted to a high carbohydrate diet. I mean, that's a population that should really be on a ketogenic diet, if not a low carb diet for sure. And that needs to be accepted and recognized if we want to help these people. I have various pharmacology students that I trained with that are trying to help them with drugs, but I think we need to take a step back and look at what's going on with their metabolic physiology. And first, the big hammer to knock their blood glucose down would be a low carb ketogenic diet. No amount of metformin or any other drug is really going to impact their disease process more than diet. And I think that needs to be recognized. And that's when getting back to the naturalness of it. I think there's different populations that it applies to. And the availability of carbohydrates 50,000 years ago, even 10,000 years ago, was really low compared to what we have now. I got a stack of books that I've read on this and lots of notes. And it ranges from up as high as 50% carbohydrates, which is pretty high in some populations, up to even 60%. But that's pretty high in fiber. But it's more likely about 20 to like 30%, up to 35%. But if you look at fossilized fecal material from people back then, there was a lot of fiber. One book really harped on this that I read recently. It was like 150 to 200 grams of fiber a day.
And the reason you're specifying fiber as being different from the rest of the carbohydrate kingdom is because that's not going to jack up your blood sugar levels.
Yeah, like indigestible fiber and insoluble, and also a little bit of soluble, but most likely insoluble fiber was part of their diet. So that needs to be recognized that our diet was profoundly different. And I think people nowadays realize that, and they're trying to figure out if that is indeed important. And I can't understand how you could rationalize that it's not important. I know there's a lot of people out there that are just advocating stick to your macronutrients, eat this macronutrient profile, and it could be high in carbohydrates, but if you just limit it and titrate it to fit your caloric needs, you'll be okay. But food is chemical energy. It produces a hormonal response in our body. And to optimize that, we really need to pay attention to what our ancestors ate and also use some of these self-quantifying technologies from a Fitbit to a glucometer to routine blood work to understand how our diet is independently affecting us, because we're really a unique metabolic entity. If we want to optimize our health, not everybody wants to optimize their performance and health like the people listening to your podcast, but if you really want to do that, you need to do sort of a little bit of work as far as blood work and self-monitoring and maybe sort of elimination diets. Like I found that certain types of nuts or dairy protein just didn't agree with me after consuming it for years. I realized that I felt better without it and certain biomarkers improved when I adjusted my diet. That could have big payoffs if you eliminate or tweak your diet and then follow that kind of optimized diet for years or decades going into what we call middle-aged, because I think we're more susceptible to diet-induced health problems as we age. So it's really good to nail down, what works for us before we get into middle age.
Let's say you don't have epilepsy. Let's say that you're really not trying to do anything therapeutic, but you would like your brain functioning as best as it can be. What is the pitch for a ketogenic diet given those parameters?
Well, first and foremost, I think they should approach it from the perspective of trying to optimize their metabolism. I view human health through the eye of metabolism. And I think if you preserve the mitochondrial function and you have very robust, highly functioning mitochondria, it's almost impossible you're going to get early onset of many types of age-related chronic diseases, even if you're at a genetic predisposition, even if you're ApoE4 positive or something like that. In that case, even though you have no outward symptoms, you want to be on a low-carb ketogenic diet. But for the average person, I would say adopt a low-carb, higher-fat, ketogenic eating strategy and see how you feel. And as you approach it with the understanding that your brain is probably hooked on glucose, and when you transition to lower carb, high fat, or even to a ketogenic diet, which is a little more severe and strict, your brain's going to go through what we call glucose withdrawal. And the best way to approach it, I think, if you're not having seizures, if you just want to approach it for health benefits, is to just do 100 grams of carbs a day is kind of a good starting point.
As a maximum.
Yeah, that'll be feeding your brain sort of enough if you spread it across 25 grams 3 or 4 or 5 times a day or something like that and kind of hit those ranges. You'll be low carb enough that you're getting a lot of metabolic benefits and shifting your physiology to greater fat oxidation, incorporating more fat in the place of the glucose. What I will recommend to people is do 100 grams a day. Many of them are starting, 300-400 grams of carbs a day, but try 100 grams a day and kind of get your body used to that. And then every other day, or maybe every third day, eat ketogenic, which would be typically, 25 to 50 grams of fibrous carbs, and then add some MCT oil. Add some ketogenic products on the market that you could use. A Ketogenics, Prove It, Ketocana from Keto Sports, different ones on the market that you can incorporate on the days that you're ketogenic and kind of ease into or slide into a ketogenic diet. So I think of your listeners out there, it's important to recognize that as we age, we should probably titrate our carbohydrates accordingly down in that direction and probably more of a high-fat ketogenic-like diet. And what I like to call my own personal diet is a supplemented, modified ketogenic diet. And Johns Hopkins uses a modified Atkins diet, and that was sort of advanced by Eric Kosoff, who's a neurologist there. And it has replaced, in many situations, the classical ketogenic diet, which is about 85 to 90% fat. And the modified Atkins diet is like 65% fat to 70%.
That's a lot easier.
Yeah. When it comes to patient compliance, that works much better. And when it comes to the fitness community, they're really not doing the classical ketogenic diet. They're more likely doing a modified Atkins diet. The classical ketogenic diet was also really high in dairy fat. And my modified ketogenic diet is really lower in dairy fat. It may get a little bit of cream, but I've replaced a lot of the cream with coconut cream and lots of avocado and other things in place of the really high dairy fat that was classically associated with the diet. And I call it supplemented because ketogenic supplementation in the form of MCT oil, the C8 oil, or ketone salts, which we're developing and testing are on the market, can really make the diet easier for people and kind of avoid some of the keto flu symptoms too that people get initially when your body reacts in its way to this glucose withdrawal. Your brain is basically sensing that it's in kind of a stress mode and your cortisol level may spike.
So at the time of this conversation that we're having right now, I actually just finished a 7-day water fast, and I know that you've done the same. One of the things that you see happening there is your ketone bodies that are circulating in your bloodstream and your glucose levels kind of move in opposite tandem. Your glucose is dropping precipitously while your level of ketones are sort of going up to bring up the slack and make sure your brain and overall physiology have something to run on. You've just been talking a lot about exogenous ketones entering the body, so presumably there's still circulating glucose from like the days that you're eating 150 to 200 grams of carbs. Then you're getting ketone bodies that wouldn't normally be in your bloodstream at the same time as you've got decent levels of glucose. In a situation like that where you've got relatively abundant levels of both, what does the brain prefer to go for?
Yeah, that's a good question. The brain will will use whatever's available in the blood. And glucose and ketones, for the most part, are not transporter limited across the blood-brain barrier and will use both fuels quite efficiently. And there's good data to assume glucose utilization can actually be enhanced by ketones. So ketones may be influencing insulin sensitivity. They reduce reactive oxygen species production in the mitochondria by enhancing sort of electron flow through the electron transport chain. Exogenous ketones, for reasons we don't fully understand yet, we're going to do liver metabolomics to understand it, but it lowers hepatic ketogenesis. Our glucose levels in our body are really regulated by our liver, right? So the glucose that you see in our blood, some of it comes from the food we've immediately ate, but more likely it's pretty much all coming from the liver is the master regulator. And if you bolus ketogenic agents, ketone esters and salts, and to some extent MCTs, it will decrease your blood glucose levels. And I think that's fascinating. And it does it more than agents like metformin. We could get a much bigger decrease in glucose with bolusing exogenous ketones than we can with metformin. So this is something that we're really interested in because we think the therapeutic implications are really high. And it also helps people that are using the ketogenic diet for medical reasons to get into that metabolic zone, if you will, which is a glucose ketone index of like 1, which means your ketone levels are elevated in your blood to the level of glucose. So your brain is kind of getting a 50-50 fuel source. Your brain readily adapts, is metabolically flexible in such a way that it readily adapts to utilizing ketones. And in a fasted state, if we fast for up to a week to 2 weeks and 3 weeks, we can get the majority of our brain energy metabolism can be fueled off ketones. Coming back to your question, so does the brain prefer to use ketones over glucose? And I get that question a lot, and I don't know how to answer it definitively, but I know in the context of something like brain injury, where you have impaired glucose utilization in the brain, or you have Alzheimer's disease, even early onset Alzheimer's disease, your brain has impaired glucose utilization due to reduction of the GLUT3 transporter to the membrane.
Right.
Pyruvate dehydrogenase complex becomes inactivated upon brain injury. A way to circumvent that and restore brain energy metabolism is to use an alternative energy substrate like beta-hydroxybutyrate. And that instead of you have like 20 different steps from glucose to deriving ATP in the mitochondria, whereas ketones just kind of blow right through the membranes, through the monocarboxylic acid transporters, and can readily go and fuel the Krebs cycle to generate the reduced intermediates to drive the electron flow. And it does that much more efficiently than glucose.
That's sort of an interesting distinction, how the general physiology of your body will preferentially go for the glucose and kind of use all the glucose it can, take that off the top until it's kind of hitting the bottom of the barrel before it starts dipping into your adipose tissue and getting you into a state of ketosis. Whereas the brain sounds like it's a lot more equal opportunity. It'll take whatever it's got access to, whether that's glucose or ketone bodies.
Yeah, it's true. And we do need baseline level of glucose. Of course, glucose can never be zero. I think there were some studies done showing, you could sustain it for a while, but glucose contributes to a lot of biosynthetic processes, to the generation of, various neurotransmitters. Ketones do too. Ketones are very anaplerotic, which means they increase the Krebs cycle intermediates. We call that the tricarboxylic acid cycle intermediates, and generates, for example, alpha-ketoglutarate, which can then go on to glutamate, and then glutamate can be converted to GABA. And it's interesting, the ketogenic diet can sort of amp up that process and make more glutamate, but it also influences the neuropharmacology of the brain in a way that converts more of that glutamate to GABA through a greater activation of glutamic acid decarboxylase. So the GABA to glutamate ratio increases. And that may impact, I'm sort of looking at that to explain some of the anti-anxiety effects we're seeing with nutritional ketosis. And it's all these mechanisms kind of working in synergy to produce the output, which is sort of enhanced brain energy metabolism that creates a brain that's more resilient against factors associated with aging. And from my perspective, from the big hit of oxidative stress you would get from oxygen toxicity, it preserves brain energy metabolism in that context. And it does that through enhancing bioenergetic intermediates, electron flow. We know now that ketones activate increased levels of adenosine. So you have higher brain blood flow, you have some vasodilation in areas does to better couple metabolic activity with substrate and O2 utilization. And it impacts the neuropharmacology in the brain in such a way that it reduces glutamatic cytotoxicity and enhances GABAergic function. And essentially, all seizures are in some way associated with glucose dysregulation, but also glutamatic cytotoxicity. And the ketogenic diet works nicely to offset that, to mitigate some of that.
Something that occurred to me while I was doing the fast, which was, you mentioned a little bit about the painful transition people sometimes have, oftentimes called the keto flu, ketogenic flu, because it's flu-like symptoms as people are having their metabolism switch over into fat burning. But it's interesting that there doesn't really seem to be a parallel in the brain. It's like your body might feel crappy for a couple of days as you're transitioning over, but people's cognition, if anything, seems either unaffected or sometimes even helped.
Yeah, I remember on day 7 of my 7-day fast, my brain felt really lucid. Actually, during that week, I wrote several grants and worked on manuscripts and just kind of stayed at home and just took it easy for the most part.
Yeah.
But during that time, I did feel more lucid, but I felt like on day 7, I was talking slower. I was almost slurring my speech. It's like I felt fine and I felt I could function. I even gave a lecture at the end and everything and felt perfectly fine, but I just felt like the whole energy systems of my body were just kind of tuned down a little bit.
Right.
And my body temperature was down about 1 to 2 degrees. And what was interesting to me as a diver, I was just sort of playing around in the pool and I was like, let me test my breath hold time. And my breath hold time increased like over 100%. It was almost like 200% higher. I was able to hold my breath close to that of people who have a lot of training. And I had absolutely no training. My wife and I do a lot of snorkeling and sort of diving down, and we'll go down to 30, 40 feet, sometimes 50 feet just with our snorkel stuff. So my breath hold time was over a minute, definitely, but I was able to hold it for like 3 minutes pretty easy. And that was completely, so being in a state of day 7, it's pretty extreme fasting ketosis. That was a really remarkable effect for me, and I want to go back and study it. I just haven't taken the time to do a whole 7-day fast again.
Were you already fat adapted when you went into the fast?
Sort of. I had read the book by Eric Kosoff and John Freeman from Johns Hopkins and did more of a classical ketogenic diet. But I would do it and then get off of it and just kind of stay low carb and do some blood work on that. So I was kind of testing a lot of different things, but always pretty much staying low carb. And then I was sort of inspired by the work of George Cahill from Harvard Medical School, that study that was published. First author would be Oliver Owen, who's a really interesting guy. And I was like, well, if these people fasted 40 days, I'm kind of healthy and fit. I could do 7 days and just see what happens. And when I did that, I found it to be much easier than expected, especially past the 3rd day. The 3rd day was a little bit, I just started not necessarily getting hungry and cranky, but I just missed food. Like eating for us is a social event. Like we wake up and actually have breakfast and dinner and don't have a 3rd meal. But it's like we sit down, and just have kind of talk and everything. And I kind of missed that whole putting food in your mouth effect.
Having done it twice now, I think the psychology of it is actually a lot harder than the stomach rumbling.
Yeah, exactly. But the most profound thing to me was that my mental capacity was very clear. I could focus more, not so much initially, but probably at a heightened state of focus. And my brain was pretty calm. But I don't know if everyone would adapt like I adapted. And sometimes I think women, quite a few people have emailed me, and even sort of in my immediate environment, some females wanted to do the fasting thing, and they sort of crumbled at like 36 hours into it. 1 or 2 of them fainted even.
That's really interesting. I've seen that anecdotally, like within the groups. Both times I've done it with a small group of people who are listeners to this podcast. I just said, hey, I'm going to do it on this week. Anybody that wants to do it with me, let me know. And it has seemed to be a harder ride for the women. And I think like when people tap out, I think in general, women have tapped out earlier.
I think women's physiology are much more sensitive to perturbations and glucose. So a woman will have a much greater anxiety response and maybe even sympathetic response to hypoglycemia. And I think that there's, evolutionary reasons for that. But I think at least in athletes, a woman who's an athlete probably bounces in and out of ketosis or hypoglycemia naturally, because there's a phenomenon called post-exercise ketosis if they do prolonged exercise. And if they're adapted to sort of that intermittent bouncing in and out of ketosis, they will probably adapt much easier to fasting.
Yeah.
But I think women may have a different sort of metabolic adaptations. And the National Institutes of Health Health is actually asking that all investigators not only just study males, because most basic science research is really biased towards males because it eliminates the estrous cycle, which can throw a huge variable.
Yeah.
And we've been guilty of doing that, but now we're looking at males and females equally. And basically what comes out of the data is that you just get more variability with females and a greater variability of responses. But I think a diet sort of needs to be adjusted in a way for females. And I think there's various factors involved, maybe even fatty acids. When people throw together a ketogenic diet, many of even clinicians and dietitians do this. They just look at macronutrient ratios, X amount of fat, X amount of protein. But I think the array of fatty acids in the diet is equally as important. So you don't want it all like omega-6 fatty acids or all saturated. You want some balance of monounsaturated essential fatty acids, because these fatty acids have important brain stabilizing effects and even neurohormonal effects that we need to take into account. And I think if those things are optimized and the diet is sort of optimized in that way, and we don't really know what's optimal yet, I think we can make this process a little bit easier, at least for following the ketogenic diet. Fasting, I'm not so sure.
If people are going to be following their blood numbers and they want to be able to safely say that I'm definitely making the dictionary definition of ketosis, factory approved by Dr. Dominic D'Agostino, what number should they be shooting for? And here we're talking about millimoles per liter of blood. Those are the units that we're talking about when we're talking about the blood prick tests for circulating ketone bodies. I think it's a different unit when you're doing the P strips.
I like the glucose ketone index, and that's convenient for some people, but it still involves, we don't have a meter yet that just reads the glucose ketone index. But I think if you're at 1 millimolar, you're pretty much like 10 times higher than you would be otherwise. So that's a significant amount of substrate in your blood, and that's providing your brain with, an alternative energy substrate that is definitely having an impact on brain energy metabolism. So that would be sort of like the lower end of it. Anything above 0.5 millimolar is technically clinically in ketosis.
Right.
With the blood measurement. What I actually recommend to people who want to save a little bit of money, first buy some cheap keto sticks at your Walgreens or CVS pharmacy, right? And they're like,
these are the ones you pee on?
Yeah, and you buy their brand, it's like 25 cents a stick, if not even that. And first confirm that you're even in ketosis, and then once with the urine sticks, and then you'll get something on the meter at least. So if you're hitting 15 to 40 milligrams per deciliter, I think in the urine strips, then you're going to be hitting well above 0.5 on the blood meter.
Donate some urine before you donate any blood.
Yeah, so you'll be getting a reading, at least, and getting a positive reading on that. And I think as you fast, usually 24 hours of fasting will get people into that 1 millimolar range. And then once you're on day 2 and day 3, you'll start approaching 2 and maybe 3, depending on the individual. And I've seen people fast for like 3 days and even still be at 0.5.
Wow, really?
And I've seen other people be at 5.2 at 2 days into it. So it's highly variable.
What are the things other than what you're actually eating that affect the levels of circulating ketones? I imagine exercise probably. Is there a circadian rhythm element to it?
Yeah, I think there is. When we wake up in the morning, our cortisol's higher. We probably have higher rates of gluconeogenesis in the morning. And I think that factors into it. Most people's blood ketones are lower in the morning, and then they sort of become elevated throughout the day and peak mid-afternoon to early evening. And that's the case with most people that I've talked with. And I think it sort of couples with a lot of our hormones too. When we wake up in the morning, I think our sympathetic nervous system sort of becomes activated and cortisol's higher. And I think that may be part of it. But we can stimulate the process through various factors. Prolonged exercise. If someone's having a hard time getting into ketosis, doing 2 to 3 hours of prolonged exercise to deplete liver glycogen will help get them there.
Right.
Eating like a fat bomb will help get them there. So eating a meal that's 90% fat or even 100% fat can help stimulate that process. Various stimulants can do that. People have showed me data showing that if they take ephedrine or other, even caffeine and other things can help. Caffeine in moderation. I think too much caffeine can maybe throw you out and create sort of a sympathetic response. But caffeine does mobilize fatty acids from adipose. So you have the greater fatty acid fat flow you have to the liver, or just in general, the more you're going to be creating ketones.
Somebody was asking me about this on one of the first days of the fast, and my answer was, I think yes, but I'd love to get your thoughts. If somebody sits in an ice water bath and just cools their body temperature forces themselves to burn sugars, I guess, at the beginning to heat themselves up and keep their core temperature up. Is that another way of sort of slingshotting into ketosis?
Yeah, I think so. I think that would definitely work. So when I was an undergrad student, I did a hypothermia study where they put me in cold water, and I had like a thermometer up the other end, and they were pulling blood from me like every minute and looking at my catecholamines. Then they threw me into a cold room and did the same thing, and they actually shared the data with me. So my blood glucose actually went up kind of significantly, as did my catecholamines, and then it would come back down. And I wasn't adapted to it. I wasn't even like a scuba diver at the time, so I wasn't even used to going into cold water, but I was shaking all day. Like I had this massive sympathetic adrenaline dump that was just stimulated all day, and that will stimulate a lot of glycogen depletion in the liver. And it's the glycogen levels in the liver that really will dictate ketogenesis to some degree, and also the hormone insulin too. And if the insulin-glucagon ratio is lower, you have more glucagon and less insulin, that really drives beta oxidation of fats in the liver and more ketone production. And that's kind of what's happening if you jump into cold water or an ice bath. So I would be interested if someone has data on that with and without, if they start a fast and they initiate it in an ice bath. I would like to see that. I would like to see not only the ketones, but the blood glucose response to that. I found that getting out in the sun really stimulates fat metabolism and really stimulates my ketone production. So laying out in the sun, I guess in Florida, it's like basically saying you're going into a sauna with UV exposure, right? I've got my blood glucose in the 40s doing that, just following a ketogenic diet. And if I'm inside, maybe I'm 65, 75, and I can go out in the sun and just, especially if I'm in a really relaxed state, and our backyard is kind of like nature. There's a little lake and geese and birds and turtles and things like that.
Yeah.
And if I'm really relaxed state in the sun, my glucose takes a big dip and my ketones go up. So that's sort of evidence that sunlight is one way or another sort of stimulating metabolism to enhance ketosis. And I know there's a lot of chatter on social media now about the effects of sun and light and UV light, and I'm starting to believe that there's really a big effect on metabolic physiology of being out in the sun. And obviously it's hard to delineate or separate the effects of heat, which going out in the sun is producing, and the sunlight exposure too, because you're sweating. It's almost like a sauna. But I've done quite a lot of blood work on myself to show that could help get you into that metabolic zone. So if someone's fasting, it would be good for them to sort of get outside, get out in the sun, maybe expose themselves to some heat, and that would initiate the process faster.
Right.
Some people will say, just can't get into ketosis, or I can't get my ketones elevated. But I think definitely some tricks out there that could push you in that direction to make it easier.
I felt a distinctly different mind state in my last couple of days on this acaloric water fast than I really ever did when I was just eating ketogenic for a while last year. And I'm wondering if maybe that's because 7 days into the water fast with a presumably as high a level of ketone bodies to glucose as I'm ever going to have, higher than I would have on a ketogenic diet. Is that maybe some of the difference for the difference in kind of perceptual feel there, or do we really know?
Well, I think in a state of hypoglycemia, you're activating many other things, like AMP kinase is activated, mTOR is suppressed, and you're probably increasing the permeability of ketone bodies, increasing the permeability across membranes by enhancing monocarboxylic acid transporter localization to the membranes and just transport into the mitochondria more. So you're probably utilizing ketones a little better than you would be on a pure ketogenic diet. So the data that I've seen, and this comes from Stephen Cunnane, he's in Canada and done some interesting research looking at the effects using a ketone PET scan. So instead of a fluorodeoxyglucose PET scan, he has labeled ketone PET scan. And the calculations and predictions that I saw was that for every millimolar of ketones in the blood, that would account for about 10%. So 1 millimolar is about a 10% increase in substrate to the brain, and 2 would be 20, and 5 millimolar, which you get during fasting or a very strict ketogenic diet, would be supplying 50% of the brain energy. So that's from some of the hard data that I've seen on that. But even if you're at 1 millimolar, 10% increase in energy to the brain, that's not insignificant. That's a pretty big boost. And it's also probably impacting dynamics that we don't fully have a handle on yet.
Right.
Like the ketones may be stimulating anaplerotic reactions that are enhancing TCA cycle intermediates. We know that that's changing the Krebs cycle intermediates. So there are things states that are being changed, not just brain energy metabolism, not just substrate. There's also neuropharmacological changes and hormonal changes that may be contributing to some of the things that you're experiencing.
Yeah.
And even like, your parasympathetic to sympathetic nervous system is changing. Like you're probably becoming more parasympathetic dominant and shutting down. Fasting can sort of shut down your sympathetic nervous system. Actually, in the beginning, it may increase it because it's a stress response. And then over time, your sympathetic nervous system starts to shut down and your body temperature even comes down a little bit to conserve energy. So you might be experiencing some of that.
Yeah, I think the perceptual difference between the first couple of days and the last couple of days is pretty profound and sort of in opposite directions. And when you think about what a situation would be for a starving animal, it kind of makes sense because for the first day or two when you're starving, it's like, well, you got to work harder. You got to find some damn food. But if you're still starving after 4 or 5 days, it's kind of like, well, whatever you're trying has clearly failed. And so it's almost like you need to stay still and conserve your strength, or at least do some real deep thinking and come up with a different strategy. I was feeling both clear-headed and fairly relaxed and expansive in my thoughts towards days 5 through 7. And I was sort of trying to tease apart why that might be, and that's my best pop psychology version of an explanation.
Yeah, I think you're well adapted. And I think failure to cognitively adapt to prolonged fasting would not be evolutionarily favorable, right? So if we were poorly cognitively adapted to fasting, we wouldn't be able to forage and acquire the food resources that we needed for survival. So those animals that couldn't do that didn't live on. So I think humans are definitely hardwired to benefit from this state of fasting. And I think it enhanced our ability, perhaps our mental lucidity, to be able to forage, acquire food resources. So I think what you're experiencing probably has evolutionary origins in that way. Maybe females are not so kind of in tuned to that because they weren't having to go long distances to hunt larger animals.
Right.
But early man, from what I garner from reading a bunch of different books, really kind of benefited from a lot of small game too. Digging, foraging, getting insects were actually pretty high on the menu in some areas. So, and it takes some strategies to acquire a sufficient amount of insects to meet your energetic needs. And that kind of nutrition is interesting to me just in traveling different places recently in Southeast Asia and other areas where insects are on the menu.
Yeah, you go to Thailand, there's some big bugs there.
Yeah, even tarantulas. I was in Cambodia. Tarantulas are on the menu. I was in Hungary recently. Brains are it's a delicacy to eat brains in many parts of the world. So I had some fried brains with eggs. And brains are like astronomically high in DHA, docosahexaenoic acid, and other nutrients. I mean, brains are literally brain food. You probably get some benefits from not overly cooking them. So they were lightly cooked in butter and coconut oil, and they tasted really, really good.
Fasting, longer-term fasting, like the 7-day fast we've done, intermittent fasting, and ketogenic diets are sort of related ideas that get tossed around together a bunch. Are there additive benefits for somebody, say, doing a ketogenic diet and then doing intermittent fasting on top of that? Or does doing one kind of get you out of the benefits from doing the other?
Yeah, I think they're very complementary. And I think intermittent fasting is definitely much easier if you're doing ketogenic intermittent fasting, right? Because say if you're doing a high carb intermittent fasting and you eat between the window, say you do a work day and you eat between 4 p.m. and 10 p.m. and then you go to bed and you eat carbohydrates, you're going to get a pretty big insulin spike and you're going to wake up probably prematurely from hypoglycemia, really, especially if you're an insulin sensitive person. That's what happens to me, actually. If I go off my diet and eat carbohydrates or too many, a little bit of carbohydrates is fine. I kind of get most of my carbohydrates at night, actually, but it's pretty high in fiber. You're going to wake up hungry and it's going to be harder, and you got to kind of suck it up and get through that hunger in the morning to get through the day before you eat again at night if you do sort of that kind of intermittent eating pattern. So if your brain and your body is keto adapted, then that morning, the fasting part of the intermittent fasting, the 20 or 18 hours, is going to be much more easier for most people if you do low carb or ketogenic intermittent fasting. And I think some of the benefits that you get in regards to activation of AMP kinase, suppression of mTOR, enhancement of even insulin insulin signaling may be a little bit better. But if you're doing pure intermittent fasting, it might be okay to get in even 50 to 100 grams of carbs and be perfectly fine with that. I just think there are benefits, complementary benefits to combining the two.
Keeping on the theme of supplementation, one of the things that I've been doing reading about, I know that salt is something that we sort of need to have a different relationship with when we're eating ketogenic than we otherwise do. And salt supplementation is something that seems pretty important. How much salt does a person need? And is there any real upside to going with one of these exotic sounding salts like pink Himalayan rock salt versus just iodized table salt?
That's a good question. I haven't seen any data to convince me that sea salt, Himalayan sea salt, would be better than iodized salt. It makes theoretical sense that it should be, but I think most importantly, you want to ensure that your body has the minerals, the sodium, potassium, magnesium, and calcium that you need. And the sea salt may be a better option because it has a lot of those minerals in it, but so do food does too. But most important thing would really be sodium. And I know sodium gets demonized a lot, but I personally get about 4 to 8 grams of sodium a day. And I'm testing various ketone products that have a sodium beta hydroxybutyrate in it.
And that's like 2 to 3 teaspoons, is that about right?
Yeah, roughly for 6 grams a day of total sodium. But when it comes to salt, I don't know if there's any, major benefit. Just adequate sodium is important. And I think if your sodium's too low, that's much more dangerous than too high, unless you have some kind of pre-existing kidney disease. You want to make sure that you're getting ample amounts of sodium, especially on a ketogenic diet, because, and especially when you're fasting too, the suppression of the hormone insulin causes your body to release a lot of sodium. And if we spike insulin with like a sugary meal or bread or processed carbohydrates, that spike in insulin will cause our body to retain a lot of sodium. That's why you blow up after going out to eat like a Mexican food place or something.
And that's why in the first couple of days fasting, you lose all that water weight because you start excreting sodium and then the water comes off with it.
You dump a lot of sodium. A lot of that's due to your insulin just goes way down. I got my insulin measured in a fast. It was like non-detectable. And I did it on a calorie-restricted ketogenic diet, and it was below the reference range. It was like almost halfway to the normal reference range. My IGF-1 was also really, really low. And that's going to cause your body to dump a lot of sodium. So I think it's even more important to ignore the dietary guidelines and to make sure that you're getting adequate sodium to preserve your fluid volume. Because once you become hypovolemic, that can cause a headache. That can cause all the symptoms that people associate with the ketogenic diet. Like you just become sluggish, you get a headache, you have orthostatic hypotension. If you're laying down or sitting and then you stand up, you get a little bit dizzy. So that's a good indication that your fluid volume is low. So especially in the beginning, you want to ensure that you get an adequate and at least double the amount of sodium that the dietary guidelines, so 5 or 6 grams of sodium would be appropriate, and lots of fluids. And your renal metabolic / renal physiology is very adaptable. So you will adapt over time and start to conserve some of that sodium. So it's not like you're going around, if you transition to a ketogenic diet, it's not like you're going to be sort of dehydrated, hyponatremic all the time. Your body will adjust over time. But that adjustment, it can be sluggish in other people. One thing that also benefited me tremendously was supplemental magnesium. And if you look at the blood work of people that are on the ketogenic diet or fasting for prolonged periods, their magnesium levels are kind of low. And the magnesium supplementation is great, especially right before bed. And it's like magnesium citrate glycinate mix, and it comes with like citric acid powder. And I drink like a gram of it before bed, and it really calms me down and I get great sleep. So I would recommend that to your listeners, some kind of magnesium supplementation.
One side effect that you didn't mention there in your list, which hopefully means that there's a way around it, is the ketone breath, the smell of acetone on the breath. That I was definitely getting that during my fast. I'm hoping that there's a way to not have that be just the new normal.
Yeah, I remember having that originally, and maybe I'm just used to it now, but I don't recognize it in myself so much.
Well, what does your wife say? That's the real question.
She thinks I'm paranoid about it, and I always ask her, it's like, no, I don't smell anything. She's pretty honest too. Like if something's off, she would definitely tell me. But I do recognize it in some other people that I've met that are on the ketogenic diet and have been for a long period of time. I will tend to chew gum in the middle of the day sometimes, like maybe twice a week. I'll actually chew a nicotine gum, like especially if I'm writing or something like that.
Right.
I have kind of like a sugar-free erythritol sweetened gum, but also hydration. Just staying hydrated can cure a lot of bad breath. Also, coffee and green tea are pretty good. And these are things that I will drink sort of in the morning, and then in the middle of the afternoon, I will brew a less strong coffee or have some tea. As long as I stay hydrated and kind of drinking things like that, I think it can neutralize neutralize some of it. But I think some people just convert more acetoacetate to acetone than others and will have a fruity breath.
Yeah.
But I found just hydration and something as simple as chewing gum can prevent that.
For somebody that's trying to keep themselves honest on keeping up in the correct millimolar range for ketosis, and I love fruit, and that's really the hard thing for me about a ketogenic diet is giving up all the fruit that I normally eat. So if I basically want to burn one of those blood strips a day and just check at some regular point where my ketones are likely to have dropped as low as they're going to based on what I'm eating, like when is the best time of day for me to check my blood ketones to keep myself honest?
Yeah, I would do that. It'd be consistent in that you do it in the same time of day, give or take like an hour or so. Mid-afternoon, I think, would be, from my own blood measurements and from seeing a lot of measurements from other people, I think midday is a good time of day to check it, like 3 or 4 in the afternoon. Just be consistent with that. And if you're 1 millimolar and above, you're doing pretty good. So it takes some pretty consistent dietary maneuvers to stay at that 1 millimolar range. That's excluding the use of like MCT or exogenous ketones, and doing that strictly through diet. And you could easily double that with an MCT supplement or one of these commercially available ketone supplements on the market. You could easily keep that at like 2 if you wanted to. And that's with a modified Atkins. It doesn't have to be the strict classical ketogenic diet. The interest in nutritional ketosis has grown tremendously since I got into this. And with all the research that I know will be published in the next year or two, I think you're going to see growing interest in using nutritional ketosis as a way to augment cognition or enhance cognitive function.
Smart Drug Smarts.
So thank you so very much to Dr. Dominic D'Agostino for taking the time for that conversation. I can't help but keep having my mind blown when I think about ketosis and just what a weird big deal it is that we have this totally secondary energy system that most of us never tap into. Now, I'm famously and unabashedly, unrepentantly a fan of fruit, which is sugary as sugary gets. Not quite the same as processed sugar, but not all that different, I guess, physiologically speaking. And I don't think I would ever go completely 100% ketogenic 12 months a year. I could see maybe weaning my fruit consumption down over a multi-decade sort of process. But I also really like the idea of doing sort of a couple months on, couple months off, maybe a seasonal thing where winters are spent in ketosis, summers are spent in more of a low glycemic index glucose metabolism phase. But given that we've all got this secondary system, I was about to call it a backup system, but I think the case could be made that maybe it should really be thought of almost as a co-primary system. The fact that so many of us never take it out for a spin when there do seem to be so many benefits, that's either a shame or a call to arms, depending on what your perspective is. But I don't think that we're nearly at the end of what we're going to be finding out over the course of the next couple of years, decades, whatever, about ketogenic diets. It seems like science is getting a late start studying this, but it's definitely a fruitful area of research. But anyway, let's move along now, as promised, to the ruthless listener retention gimmick.
Smart Drug Smarts. Ruthless listener retention gimmick.
So it's no secret that physical activity and the brain are intimately interrelated. Our brains make our bodies move, of course, and the movement of our bodies can impact the workings of our brain. The latter is sometimes a little less obvious, but recent studies have shown that many of the neuromodulating chemicals that are needed to transition memories from short-term working memory into long-term memories, the levels of those neuromodulators can be affected by physical exercise. These are things like dopamine, noradrenaline, and that acronym that everybody seems to be excited about these days, BDNF, brain-derived neurotrophic factor, which is very much affected and increased by physical exercise. But a recent study conducted in the Netherlands wanted to see if the timing of exercise relative to the time when something happened that was meant to be remembered would have any effect on the laying down of long-term memory. In other words, something happens that's worth learning, worth remembering for next time, and you're going to get some exercise. Do you do the exercise immediately after the learning? Do you do the exercise several hours later? Or maybe the exercise isn't that important after all? They ran the test. The researchers recruited 72 participants, and they tasked them with learning to match a series of 90 locations with pictures over the course of a 40-minute period. Then some of these people immediately exercised, some of the people didn't exercise for 4 hours, and a control group did not do any exercise at all. Then 2 days later, the participants returned to the lab to see how much of what they were trying to remember they actually could remember. And it turned out that exercising immediately following the learning had no effect on memory. You might as well not have exercised at all. But the group that waited for 4 hours and then exercised, they showed significantly better memory for what they had tried to learn than either of the other two groups. Guillen Fernandez, the senior author of the study and the director of the Donders Institute for Brain Cognition and Behavior, said that while strong memories will be remembered no matter what happens, it's these weaker memories that would normally be forgotten within a day or so that may last longer as a result of exercise. It's a little bit unfortunate that in the study they broke it up into just 3 groups: the people that exercised immediately, the 4-hour group, and then the not at all group. It'd be nice if they did it like hour by hour going out for the next 10 hours, but maybe this first finding is enough that other researchers will follow up on it.
We like to think of ourselves as a digital speakeasy for brain hackers, but you can call us Smart Drug Smarts.
Okay, that is all for episode number 147. Thank you for hanging around until the very end. If you have not eaten for this whole episode, we've probably depleted your brain glucose and you're well on your way towards fat metabolism. That might not be entirely accurate, but hopefully some of you might be inspired to give some ketogenic diets or something like that a try. The links to everything that we talked about here will be up online at smartdrugsmarts.com/147. If you missed last week's episode, that was a big review of nicotine, that most bad reputation of drugs, but maybe there's a silver lining there. That was episode number 146, worth checking out if you have not yet done so. And next week, I think that we're going to be having another Know Your Neurotransmitters episode, although I think technically this is a neuropeptide, not a neurotransmitter, but oxytocin, also known as the love drug, the snuggle drug, the makes moms love their baby whether they're otherwise inclined to or not type drug. Super interesting compound, and barring catastrophe or change of plans, that'll be coming up next week. Reminder that this coming Wednesday, if you catch this episode soon, we're going to be having a live Q&A session with Dr. Dominic D'Agostino, doing that on Twitter. You can check that out at smartdrugsmarts.com/axonchat for more details. And otherwise, I will see you back here next Friday, same time, same podcast, and with the same unflagging commitment to helping you fine-tune the performance of your own brain. Have a great week and stay smart.
You've been listening to the Smart Drug Smarts podcast. Visit us online at www.smartdrugsmarts.com and subscribe to our mailing list to keep your neurons buzzing with the latest in brain optimization. Smart Drug Smarts should be listened to for entertainment purposes only, although some guests on the show are medical doctors, most are not, and the host is just some random guy. Nothing you hear on this podcast or read at smartdrugsmarts.com should be considered medical advice. Consult your doctor and use some damn common sense before doing anything you think might have a lasting impact on your brain.